Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this lecture on catabolism of the carbon skeleton of amino acids, I will be concentrating on amino acids that form TCA cycle intermediates. Let's get into the details. Now, the amino acids that form TCA cycle intermediates, the first one that I would like to explain here is the asparagine and aspartate. These are some of the two amino acids that form a oxaloacetate. So the oxaloacetate is a TCA cycle intermediate. So asparagine and aspartate make oxaloacetate. You know TCA cycle. Just to recap the TCA cycle intermediate, just for your sake of understanding and for the benefit of this lecture. So we have acetyl-CoA getting into the TCA cycle, combining with oxaloacetate, and acetyl-CoA combining with oxaloacetate will make citrate, and citrate will make isocitrate. Isocitrate will be converted to α-ketoglutarate. Alpha-ketoglutarate is converted to succinyl-CoA. Succinyl-CoA converted to succinate, and succinate is converted to fumarate. Fumarate is converted to malate, and malate is converted to oxaloacetate. This is what is a TCA cycle reaction. Alright. So what we are seeing here is which amino acids upon catabolism, their carbon skeleton make oxaloacetate. We are looking at that now. So asparagine upon catabolism and aspartate upon their catabolism of carbon skeleton will make oxaloacetate, which is a TCA cycle intermediate. That's where we are now. Okay. So asparagine, which is a non-essential amino acid, aspartate, again a non-essential amino acid, can go in when they undergo catabolism. So they will give oxaloacetate. Let's see how. First, let's see how asparagine is converted to aspartate. So asparagine can undergo a deamination process done by asparaginase enzyme. The enzyme that causes deamination here is asparaginase. And this asparaginase enzyme removes that the side chain amino group, basically causing deamidation. Side chain amino group is removed as ammonium ion, and that's how asparagine is converted to aspartate by asparaginase. Once you get aspartate, aspartate undergoes transamination reaction catalyzed by AST, that is aspartate transaminase enzyme, where the amino group of aspartate will be given to α-ketoglutarate, and that α-ketoglutarate will be converted to glutamate. As you know, in all transamination reaction, in my previous lecture on transamination, I have explained you the acceptor of primary amino group during transamination is alpha-ketoglutarate, always. Alpha-ketoglutarate, after accepting the amino group from an amino acid, it will be converted to glutamate, while an amino acid, after losing the amino group to alpha-ketoglutarate, so that will be converted to its corresponding keto acid, and the corresponding keto acid here for aspartate, it is oxaloacetate. So that's how oxaloacetate is made. So with the help of two enzymes, asparagine is converted to aspartate by asparaginase enzyme deamination process, and then the aspartate transaminase enzyme converts aspartate into oxaloacetate, and the oxaloacetate, it's a TCA cycle intermediate, and oxaloacetate can be converted to malate if there is sufficient NADH available. Oxaloacetate reverses into malate. Malate moves out of mitochondria into the cytoplasm, and in the cytoplasm, malate can be malate moving out of mitochondria into cytoplasm. In the cytoplasm, malate is converted back into oxaloacetate, and oxaloacetate will form phosphoenol-pyruvate, and phosphoenol-pyruvate goes into glucose formation. That's how asparagine and aspartate are glucogenic amino acids. So overall they make a TCA cycle intermediate oxaloacetate. That's what is the purpose of this particular slide. Now, one of the application here is the asparaginase enzyme, clinical application. Asparaginase. Now the thing is, asparagine is synthesized in our cells because it's a non-essential amino acid. The synthesis of asparagine from aspartate is done by asparagine synthetase enzyme. Let me take this out. Now the asparagine synthetase enzyme, asparagine synthetase. Now the conversion of aspartate into asparagine, it needs an amino group, and the donor of amino group in that conversion is glutamine. The side chain of glutamine, which contains an amino group, side chain amino group, is given to aspartate to convert that into asparagine, while glutamine is released as glutamate. This particular reaction is catalyzed by asparagine synthetase enzyme. Okay, so most of our cells will express asparagine synthetase enzyme, so they will make their own asparagine, and that's why asparagine is non-essential amino acid. As long as we have aspartate, asparagine can be synthesized by getting a side chain amino group from glutamine. Now certain tumor cells like acute lymphoblastic leukemia tumor cells that I have written here, acute lymphoblastic leukemia tumor cells, they are deficient in asparagine synthetase enzyme. They are deficient, means they don't express asparagine synthetase enzyme. So that means acute lymphoblastic leukemia cells, they are dependent on asparagine that is coming from our diet. You know, when we consume proteins, one of the amino acid there is asparagine, which is digested, absorbed and getting into our bloodstream, and from the bloodstream, acute lymphoblastic leukemia cells will take that asparagine and use it for their own protein synthesis. So they are entirely dependent on blood-borne asparagine coming from the diet, because they are simply lacking asparagine synthetase enzyme. These type of tumors are referred as asparagine synthetase deficient tumors. Now, how to treat asparagine synthetase deficient tumors like acute lymphoblastic leukemia? The one way to do that is use enzyme asparaginase. So, asparaginase, which converts asparagine into aspartate, we have seen that it causes deamination. Asparaginase enzyme, it causes deamination of asparagine into aspartate. So, using exogenous asparaginase like synthetic asparaginase, which is marketed as L-spar. So, what it does, whenever exogenous asparaginase is given to a patient who is suffering from acute lymphoblastic leukemia, so this exogenous asparaginase in the blood is going to convert most of the asparagine into aspartate, thereby decreases the available asparagine in the blood, thereby acute lymphoblastic leukemia cells, they don't get sufficient asparagine, and that's why they won't be able to make new proteins for cellular proliferation. And that's how the severity of the disease can be brought down. What about the normal cells? Normal cells, they have their own asparagine synthetase enzyme, so they can synthesize asparagine in their cells. They are not dependent on the blood-borne asparagine, and that's one of the clinical application that we can use in this from this particular reaction, that is conversion of asparagine into aspartate. So that's about amino acid that form oxaloacetate as their catabolism of carbon skeleton. Now let's move on to see amino acids that form α-ketoglutarate as their metabolic intermediate. The carbon skeleton of amino acids that form α-ketoglutarate is glutamate, glutamine, arginine, proline and histidine. All these are the amino acids that make α-ketoglutarate as their metabolic intermediate. So those amino acids are glutamate, glutamine, arginine, proline and histidine. Now let's look into that one after the other. So the glutamine, let's start with the glutamine. Glutamine undergoes glutaminase reaction in the mitochondria, especially in the liver and kidney. Glutamine, the side chain amino group, is removed as ammonium ion by glutaminase enzyme and you get glutamate there. And then the glutamate will undergo transamination reaction to make α-ketoglutarate. And that's how glutamine will be converted into glutamate and glutamate is converted to α-ketoglutarate. So, two amino acids we have seen now, glutamine and glutamate making α-ketoglutarate. So, we have covered first two, glutamate and glutamine making α-ketoglutarate. So, two enzymes here. One is glutaminase, which is causing deamination process where ammonium ion is released, and this ammonium ion can go into urea cycle if it happens in the liver. Then the glutamate undergoes transamination reaction. Any transaminases, it can be, it is here in this slide it is shown oxaloacetate converted to aspartate, or it can be any amino acid. Okay, because alpha-ketoglutarate glutamate to alpha-ketoglutarate is common in most of the transamination reactions. So we are going down, glutamine to glutamate, glutamate to alpha-ketoglutarate. Going from below to top, like alpha-ketoglutarate to glutamate, glutamate to glutamine is a synthesis of these amino acids. So we are not looking into that. We are not looking at the synthesis of amino acids here. So because we have already seen that, right? Glutamate dehydrogenase, glutamine synthetase, we have seen that in fate of amino group nitrogen, transamination reaction and all that. Anyway, so these are the two enzymes that will convert glutamine and glutamate to alpha-ketoglutarate, that is glutaminase and glutamate dehydrogenase. And also I have explained you what is the importance of glutamate dehydrogenase in the transamination reaction also. Now, let's move on to see how arginine and proline will be converted to alpha-ketoglutarate. So the proline and arginine, they are converted to glutamate gamma semialdehyde initially, and then this glutamate gamma semialdehyde, it will be oxidized to glutamate, and glutamate undergoes oxidative deamination process to become alpha-ketoglutarate, or glutamate can undergo transamination reaction to become alpha-ketoglutarate. So there are two reactions there. Okay. So let's see how proline goes into glutamate gamma semialdehyde and glutamate, and then how glutamate goes into alpha-ketoglutarate. So proline it is oxidized into pyrroline 5-carboxylate, and then that will be broken down into glutamate gamma semialdehyde. And then this glutamate gamma semialdehyde will be oxidized into glutamate, and the glutamate now it can undergo two types of reaction. It can undergo glutamate dehydrogenase reaction, removing ammonium ion NH4 plus to become alpha-ketoglutarate. That's how alpha-ketoglutarate is made. Or glutamate undergoes transamination reaction. Okay, so to transfer its amino group to any other amino acid and become alpha-ketoglutarate. So transamination reaction, transamination. So in transamination reaction also glutamate can become alpha-ketoglutarate. So overall, once you get glutamate, it will form alpha-ketoglutarate. Now what about the arginine side? Arginine undergoes arginase reaction in the urea cycle. Enzyme here is arginase. arginase enzyme breaks arginine into ornithine and urea, and that's how urea is made in urea cycle. So arginine undergoes arginase reaction in the urea cycle to make urea and ornithine, and the ornithine undergoes transamination to make glutamate gamma semialdehyde, and this glutamate gamma semialdehyde making glutamate will become alpha-ketoglutarate, which is a TCA cycle intermediate. Like this, proline and arginine ultimately be converted to alpha-ketoglutarate. Now coming to how histidine is converted into alpha-ketoglutarate. Now in this particular flow pathway, histidine undergoes histidinase enzyme reaction where ammonia is released. Basically it's a deamination of histidine into urocanate. Histidine undergoes direct deamination reaction by histidase enzyme to become urocanate. Now the urocanate undergoes urocanase reaction to become imidazolone propionate, and the imidazolone propionate undergoes hydrolase catalyzed reaction to become formaminoglutamate, FIGLU. FIGLU is formaminoglutamate, and this formaminoglutamate undergoes formaminotransferase reaction where tetrahydrofolate entering into the reaction and going out as formaminotetrahydrofolate, and glutamate is made. So this is how a histidine is involved in the formation of a 1-carbon intermediate of tetrahydrofolate derivative, that is formaminotetrahydrofolate, and this formaminotetrahydrofolate can undergo 1-carbon metabolism. That's how tetrahydrofolate is converted to formaminotetrahydrofolate. Okay, so here we have N5 formal tetrahydrofolate, N10 formal tetrahydrofolate, things like that, right? So that's how histidine makes this special type of tetrahydrofolate derivatives. Now the thing is, conversion of FIGLU, it needs tetrahydrofolate. That's the point here. As you see in this reaction, pay special attention to this reaction. So there is a requirement of tetrahydrofolate in the conversion of FIGLU to glutamate. This is the last step in the histidine catabolism, where histidine is ultimately giving its carbon as glutamate out of the reaction, and this glutamate, it can go towards alpha-ketoglutarate formation. Glutamate can be converted into alpha-ketoglutarate. That's done by glutamate dehydrogenase enzyme. So that's how histidine can make alpha-ketoglutarate. That's our purpose of the lecture. Now some of the application here, like in the conversion of histidine into alpha-ketoglutarate, in the last reaction where FIGLU is converted to glutamate, where there is a requirement of tetrahydrofolate. So what if there is a deficiency of tetrahydrofolate? Let's do that. So I'm going to decrease tetrahydrofolate now. So folic acid deficiency or the tetrahydrofolate deficiency. Folic acid makes tetrahydrofolate. So deficiency of folic acid, what will happen? So this formaminotransferase reaction will decrease. That means there will be increase in formaminoglutamate. So FIGLU will increase, and this will appear in urine. So urine FIGLU levels will be high. That is, formaminoglutamate levels will be high in the urine sample. That indicates there is a decrease in tetrahydrofolate. So using histidine supplementation, whenever histidine loading test, whenever we give more histidine, so in order to conduct histidine catabolism completely to alpha-ketoglutarate, we need tetrahydrofolate. So if there is a tetrahydrofolate deficiency, whenever histidine loading is done, so there will be rise in FIGLU because the last reaction don't go on. So elevation of FIGLU in the urinary sample when histidine loading is done, so excess FIGLU in the urine sample is indirect indication of tetrahydrofolate or folic acid deficiency. That's one of the application that you must remember. Okay. So here in this slide I have given briefly things that I have explained in the previous slide. So histidine undergoes deamination and hydration reaction to form 𝐸𝐵𝐴minoglutamate, which is FIGLU. FIGLU donates its 𝐸𝐵𝐴mino group to tetrahydrofolate to make 𝐸𝐵𝐴minotetrahydrofolate and glutamate. Glutamate undergoes oxidative deamination to make α-ketoglutarate. Alright, now let's see a FIGLU test. What is FIGLU test here? FIGLU test is done in cases of folic acid deficiency. So in folic acid deficiency, metabolism of histidine will stop at the last reaction what we have seen in the previous slide, that is it stops at FIGLU, 𝐸𝐵𝐴minoglutamate. Because conversion of 𝐸𝐵𝐴minoglutamate into glutamate needs tetrahydrofolate. So deficiency of tetrahydrofolate do not allow this conversion. So how the FIGLU test is done? So oral load of histidine is given, and after that, after some time, a plasma concentration of FIGLU and its urinary excretion is measured. And if plasma levels of FIGLU and the urinary levels of FIGLU, if they are elevated, it is an indication that folic acid is deficient. Unless there is a genetic mutation in the gene coding for 𝐸𝐵𝐴minotransferase enzyme, which is rare. So folic acid deficiency will, in cases of folic acid deficiency, after oral histidine, there will be rise in FIGLU, and that is what is an indication of folic acid deficiency indirectly. So that's what is the application of FIGLU test there. So this is all about catabolism of carbon skeleton of amino acids, especially the ones that makes a TCA cycle intermediates, and we have covered oxaloacetate and α-ketoglutarate specifically. Thank you.